Okay, thank you very much, Sonalika, and welcome everyone to this webinar. I hope that everyone found all of the details necessary for today's session. And what I will do in my part of the presentation is speak a little bit about why we are doing what we are doing, then what is the Research Fellowship Program all about, and how you will be able to benefit from different resources that we have prepared for this program, and how you can gain access to the resources that we will discuss. So again, thank you, Sonalika, for the introduction. I'll briefly go over some of the similar things that Sonalika already mentioned, but I want to specify why we are doing what we are doing first, and talk about how the team members that we have in our team will be able to help you develop research projects over the summer. So the background of our company is that we started as the technology transfer company, started from this bioinformatics research center that is focused on developing of novel algorithms to process and integrate multi-omics data. And so what our team does is leverages the same tools and develops training resources to support research projects and apply the different methods that are being developed at the center, as well as the other methods that are being tested and benchmarked to a variety of different biomedical research projects. And one of our main goals as a company is to democratize bioinformatics. And so we've been working with many different companies, universities, organizations, nonprofit organizations, as well as for-profit organizations. And the goal is really to make bioinformatics more accessible, because what we have discovered is that many students, as well as faculty and researchers coming from a life science background, do not have enough resources and training to translate research questions into data science problem statements and into a bioinformatics workflow. And so what we've been doing with a number of these partners is developing examples and case studies around different types of data and different types of challenges that arise with that data, so that students and faculty can effectively address those using bioinformatics tools. And so that's how this OmicsLogic bioinformatics training curriculum was born. We essentially developed it addressing this need of growing amounts of data and limited opportunities for people around the world to be able to easily access that data using relevant tools. And so we developed regional offices with an office in India that serves many different universities there. Starting in January 2019, we've conducted many different online and in-person workshops and programs. We also recently established an office in Africa, where we have a regional team based out of Nigeria. So I see many of you here have joined us from Africa, so welcome to all of you as well. There we are growing the impact of the program by offering these programs together with our collaborators and analyzing the data to be addressing the different types of needs that are unique to this region, and developing projects that are relevant to people in Africa as well. And of course, in the US, we have the privilege of working with a number of partners, including one of our main collaborations with the Louisiana Biomedical Research Network, as well as Georgetown University, where we have programs for students that apply for biomedical research projects and for medical studies as well. And so as a result of these collaborations, we've seen a number of students and faculty work together on impactful and translational projects. And so we're excited to bring this opportunity to you all online as well. And so what we are doing with this curriculum and with support elements that I will discuss here briefly, we are trying to enable teams of students and independent students to develop impactful and competitive research projects, leveraging big data sets, mentorships, and expert feedback so that those projects are more competitive. And so the way that this process starts is really from training. And so before we jump into the research part of this program, I want to briefly mention what this training is all about. So Narika, do we have the poll ready?
Yes, Ilya, we have it ready.
Okay, so before we go on, I wanted to make sure that we know who is present in this session today. And so we told you a little bit about our team. So Narika, please launch the poll. If you could please take a few moments of your time and answer some of these questions that will be popping up on the screen in just a second. Okay, so, okay, can we launch the poll?
So my first question is, what is your educational background? Please let us understand from which educational background you are. And then my second question is, what are your research interests? It would be great if we could know what your research interests are. Either it is oncology, omics, data science, biomedical science, machine learning, virology, R and Python programming, agri-bioinformatics, precision medicine, or neuroscience. You can take a minute. I would be ending the poll in the minute. Thank you.
Okay, so can everybody see the poll? Okay, great. All right, perfect. So if you could please answer those questions, we'll just give another minute for everybody to be able to answer. And so what we're looking for is to really get to know you and understand better what are your interests, what is your background, and also try to adapt this program to the needs of those that are present today. We also have this webinar available as a recording after this session is over. So for everyone that is watching this on YouTube, I think it's streaming live there as well. We don't have a poll available on YouTube, but you can definitely answer your questions in the chat. We're monitoring the chat, and so we'll be collecting your feedback there as well. Okay, let's give it just another 30 seconds, and maybe Sonalika, you can let me know when people have completed that.
Okay, sure, Ilya. Thank you all for your responses. Those who are left, please fill in the responses. Thank you.
Okay, so again, thank you for the responses. Maybe we can keep it on just for a few more seconds, but let me tell you briefly what is the objective of the first stage of this program. So as was mentioned, we're talking here about the research fellowship. We'll get to the research part later, but first let me explain how the basic training works. So we have an online portal that is called OmicsLogic. So you can all take a look at the portal right now. It's learn.omicslogic.com. And what this portal offers is a series of coursework. Okay, so that's the link on the basics of bioinformatics. So anyone that is present here today that just has some basic understanding of bioinformatics, or even with very limited understanding of bioinformatics, what it is and how it applies to different biomedical research challenges, we offer a basic introduction to bioinformatics. We have different omics-specific types of training like genomics, transcriptomics, metagenomics, and epigenomics. And then there is an extensive curriculum on data science that includes data exploration, data analysis, visualization, and machine learning. So please explore the different courses. Again, and Sonalika, maybe you could post a few links there so people can take a look. Now, on top of this asynchronous type of coursework, we also offer an in-depth and expert-guided training. And so as an example, we have these programs that are called OmicsLogic Transcriptomics, OmicsLogic Data Science. So we have a number of programs that are designed around the specific challenges associated with these types of data, as well as specialization tracks. And those specialization tracks include oncology, infectious diseases, agriculture, and others. And so as a result, this portal offers a complete roadmap for bioinformatics. And we go from finding data sets to knowing how to explore and visualize the data to understand the problem statement and what they represent, to read through research articles and understand the research methods, as well as to gain biological and translational insights and understanding of what is the impact of this particular method, study, data set. And so that type of training really introduces you into a bioinformatics roadmap from where you can then choose to specialize in specific domains. So the OmicsLogic Bioinformatics training is a topics and specialized program for different types of research challenges. And those include challenges in infectious diseases like COVID, challenges in genomic medicine like personalized treatment of cancer, metagenomics, how to explore the microbiome and the impact of microbiome on different aspects of disease and health, precision oncology, data science, and neuroscience. So to really explain how this works, I want to just briefly show you what kinds of materials you will gain access by joining this program, and also by just registering for free. Even if you're not registered for the research fellowship or one of these programs, I just want to make sure that you understand how these resources could be used. So they include training materials that you can learn from, data analysis tools that include big data analysis tools, because not always can you analyze data on your laptop. You really need this infrastructure to support your big data analysis needs. Training in coding, where you can try different types of R packages and Python, as well as an introduction to different project data sets. So you can really explore a variety of case studies of how this applies. So let's take a look. I'm going to go to the browser, and please join me as well. So learn OmicsLogic.com. And please do let me know if you're already registered. Maybe you could put yes or no in the chat box if you're registered. All right, excellent, thank you. So as you're on this home screen, if you're already in the system, you will see your email address come up right here. So for those of you that are not yet registered, this is free registration, so really no obligation. This is just to explore. But you can see here how some of these resources are organized, and they are specific to different types of applications: oncology, infectious diseases, neuroscience, agriculture. And what's important is that they offer, once you register, once you've created that first registration, you automatically get access to our big data analysis platform. Now, this big data analysis platform is a complete separate beast, so to say, right? It's got a lot of resources that you can leverage, which are explained in some of the coursework that you can gain access to. So let's take a look at what this big data analysis platform allows us to do. Now, you will, if you're registered on the Learn portal, you can use the same registration to see what this big data analysis platform is all about, because you will have a demo account. Okay, so this is the link to this platform. It is server.tbio.info. Okay, please let me know once you're here. Okay, so again, log in, use the same login, same email and password that you created there. You should be able to register here. And we'll take a look at just one example. So here we have different types of omics data types: next generation sequencing data, mass spec, structural biology, as well as data integration and modeling. So some of you might be already familiar with this, right? One of the biggest challenges that so many data sets are available, but they contain thousands of samples, gigabytes of data. You have to download them on your computer. Most of the time, you can't even do anything with them because they just require terabytes of space. And so this is a cloud-based platform that allows you to eliminate that challenge. You can just simply go online, upload your data directly from NCBI or from an FTP link, and process that data. And so that's one of the amazing things that are available on this platform. So let's take a look at one example. Here we have a demo of how this platform works. So you first of all start by selecting one of the demos. You have a brief explanation of what the basic steps are. So this is the logic behind this type of omics data processing. You can see that you do pre-processing, mapping on the reference genome, and then quantification of expression level. So how many of you here are familiar with RNA-seq? Is anyone here familiar? Okay, so I see Kanak Valley. Okay, Lee. All right, great. So, all right, great. So you will probably already recognize many of the steps that are mentioned here. And what you can do is not worry about the organization of these steps in a logical workflow, because the system is guided by artificial intelligence to suggest the right type of next step based on the data type and your objective. Okay, so if you are just coming from a theoretical background, you kind of know what is going on, but you don't know how practically it works, this is exactly what you can use. So remember, the first step was pre-processing. So we'll select Trim-O-Matic, which is designed to trim the reads and clean up our source data. Then we can eliminate the bridge amplification, because sequencing includes PCR amplification that is a little bit biased towards some sequences and higher expressed genes. Then we can choose between a number of different methods for mapping on the full genome or the transcriptome. So we'll just choose a quicker version, mapping on the transcriptome, and then preparing that table of expression that we will be able to use downstream analysis tools to annotate, explore, analyze, and ultimately interpret. So here we have the RSEM expression table. Once we have built our pipeline, just click on End. And now you can see the output data. And so very quickly, you can go from understanding the project to then being able to explore the data that is already processed. And so now this data is something that you can download onto your computer, or even better, I will show you how you can process it using some of the data exploration tools that we have on the OmicsLogic Learn platform. Okay, so let's go back to the Learn portal, and I'll show you the next example. So here we built our table of expression, right? So in the table of expression, we prepared a full table of gene expression for something like tens of samples, okay? So here, if you can search R, and we're going to go into this introduction to data science in R, okay? Because the next step after you've prepared your data, now you can load the data, visualize it, perform different types of statistical analysis, explore your data with dimensionality reduction, perform differential gene expression, and use all kinds of downstream methods. So here, I'll just show you a brief example of how this would work. Again, this is for those that created a free account. You should be able to access most of these courses. And what we'll do is we'll go right here into dimensionality reduction, because one of the bigger challenges is that we have so many dimensions. So here you can see we've got our genes, they have expression levels, we have samples, right? And then each sample belongs to different type of data. So this is taken from a breast cancer example project. And as you complete the introduction, you then have this video that explains what the code is all about, explains the syntax of the code so you understand how you load the data and prepare your principal component analysis object, which later on you can visualize because R is a object-oriented language. And then how can you improve your visualization using that object so that you understand the biological meaning behind the complexity of this gene expression dataset. Now, importantly, you can actually practice this right here in the browser. So again, you have this explanation, but you also have these blocks of code, okay? And this code actually comes with a challenge, okay? So for example, you can't just run this code, but you can go to a basic plot, right? So here I'm going to just run the code that's already available, okay? And I get some basic outputs. So the basic outputs include some basic plots. But once I want to start changing them, you can see that I have to modify the code for it to work, okay? Why? The reason is because I need to start reading through the syntax and identifying the specific types of steps that I need to make so that this code works. So let's briefly do this together. You can see right table, I have it right here. I need to create a table. So I'm just going to copy this from right here. And I have making the plot from the previous step. So how do we actually make the plot from the previous step? Okay, right here, we need to make the plot from the previous step. So by looking through the code and understanding the code, I can actually make this code work. Okay, what didn't work here? Let's make sure that all the syntax is true. This. Okay, and the reason is I don't have input file. Okay. Okay, and so now I made this code work. I have this code right here, and I have created a table of expression. Okay, and so now I can analyze the table of expression in a transform format, so I know what it means. Okay, so because this process is something that we've tested on thousands of users, we have developed a whole system that goes from this kind of basic training all the way into a project that you can now analyze in the context of a biological problem. And so these include examples taken from literature, where we actually go through examples in research papers with the associated data set, and you can find different examples on Ebola, on COVID-19, on precision medicine in oncology, and many other different types of examples. So in every case, you will get an introduction based on some kind of a data set that is explained in a publication. You get different types of methods being explained, and then some challenges that you can try and go through by yourself. Now, in many cases, we try to break down every step so that it's very clear what the logic is behind the analysis and not get bogged down by the technical details So eliminating the need for comprehensive technical training before you can actually start a project of your own. And so what's important is that we have this platform that allows big data processing, different types of statistical methods, regression and factor analysis, machine learning, multi-omics integration, and biological interpretation. As a result, as we mentioned, now we are going to talk about this fellowship. So who is the fellowship for? The fellowship is for those that have completed the basic training. They already understand how bioinformatics is used, and now they can apply what they have learned to a project of their own. So how many of you here have come to this webinar to learn about how to join the research fellowship? All right, great. Okay, so the research fellowship is specifically for those that are going to be able to leverage some of the examples data sets, learn about different challenges that are presented in these projects. These challenges are going to be associated with different domains, as we already described, and you can see some of the examples on the screen that we have already prepared. And once you are ready and you have selected a topic, you can now apply the same principles to a project of your own. And so you can either join the training, and I will pass it on to Dr. Mohit Mazumdar after my part of the presentation so that he can explain what kinds of programs are going to be coming up that you can gain access to expert guided training, to asynchronous coursework, and the research support that we offer. But for those of you that are done with your training and have already completed your basic training, now you can go for an independent project. And so that's where this OmicsLogic Research Fellowship comes in. So let me briefly tell you what is the Research Fellowship and how does it actually work. So the Research Fellowship is organized into 3 different parts. First, we have an introduction so that you can review and refresh your understanding of different types of bioinformatics methods, but it's going to be brief. The important thing is that as a part of this program, you will gain access to a variety of programs that we have going on during the summer so that you can get that training. Next, there's a process of evaluating the proposal, the project proposal. I'll talk about how challenging and how to address that challenge of selecting a good topic and finding a good data set. So during that time, once you have the evaluation step, you're really working on developing a research proposal to also understand the experimental design of how the data was prepared and making sure that the question that you have is actually competitive and important and significant, and you will end up with an impactful project at the end, right? So it's refinement of that research question that you have. And then you have also practical aspects so that you actually conduct the different research analysis methods, you apply them to your data, you find the insights through identifying specific patterns in the data, and then you have to go for biological interpretation. So in this process, you will have weekly progress reports that you have to submit to your mentor. You have a Slack channel where you can interact with the mentor, experts, as well as additional people that are going to be present, okay? And those of you that feel that you need training, we will talk about training here in a few minutes. But for the research fellowship, once you join, we will actually go through this process with a weekly review, and you will hear today from some of our research fellows on how they went through this process as well. So the first step, obviously, is to collect data, right? And so we will introduce you to different data sources where you can find big data on almost any topic. So many of you are probably familiar with NCBI. NCBI has millions of data sets that you can find on a huge variety of topics. And so, for example, many research data sets are available on SARS-CoV-2, right? Literally every paper that comes out has an associated data set that anyone can go in and explore. And so what we just did with that PCA for cell lines, you can just take a data set from Geo, download it, or even directly use it from NCBI and see some patterns. For example, here we have some pattern of separation based on gene expression between ACE2 overexpression in patients with asthma and with COVID-19. So you can see the difference between those two conditions. And so the obvious challenge when you are faced with millions of data sets that you could potentially access is how to choose that specific topic for your project. And so here we will provide you with some training so that you understand how to select an impactful topic, how to select a topic that is reasonable, and also how to make sure that that topic has significant data that you can analyze during your research fellowship. Then you will go through a process of literature review. So you'll get to review articles that are either primary research articles, review articles, systematic review articles, and you'll get to explore different types of problem statements, questions, and data sets that experts have already gone through. And the important part is that you will be able to define from a very general problem statement, like I want to apply my bioinformatics skills to infectious diseases, to select a very specific hypothesis or a question based on your literature review, identifying gaps and unanswered questions, and then defining your own question and hypothesis. So as a result, you will end up with a very competitive project proposal that you can actually start working on. And so then you will develop comprehension and analysis skills by going through these research articles, understanding the methodology. You will be able to investigate the problem in depth and understand what challenges are there. And then we will go to actually writing, editing, and preparing the project for submission. And so what we've done on our side, and you will see this from some of our research fellows that will present today, how this step-by-step process actually enables you to come out with a project that leverages big data, applies advanced and sophisticated methods, and then comes out as a research study that you can present and share with others. So once your data set is ready and you've defined your question, you will process your data. And so as I showed you, there's this training that will allow you to leverage these big data analysis tools and identify signal. You will be able to leverage different types of scripting methods and analysis methods to define what that pattern is that you want to focus on, perform in-depth analysis in a variety of applications from gene expression data to drug resistance and structural biology data, population genetics and others, biological interpretation like pathway analysis and others, and then start working on completing that project. And so what could be the outcome of the research fellowship? So first of all, we follow the same process that you would follow if you were preparing a research poster presentation. So as you probably know, many students start with a research poster to develop their project and get some feedback. And so this program, the Research Fellowship, will provide you with these guaranteed outcomes that, depending on the effort that you put in and the timing that you have, you will be able to achieve. And so some of these could be just understanding the challenges associated with bioinformatics. So if you're a student and you want to understand whether this is for you, you might want to go through some of the coursework, understand the challenges, complete some of the projects that we offer, and that will give you a clear perspective on whether bioinformatics is for you or how could you use it in your domain. Another outcome is that you will get training and develop those skills. And so once you've tried it on one project, you can then continue working on other projects and apply the same skills and the training that you've gotten so that you can actually understand how this is applied in a variety of different areas. Then you can work on an independent or a group research project. And so that's where this 3-month or 6-month training converts into a research fellowship that you can actually work on a project either independently, or we can match you with some similar people that could be interested in working together. Okay, so you will fill out your profile. We'll see how you do in training. You'll submit some of the research progress that you're making, and then we'll be able to pair you with some others. And then finally, what we definitely see majority of the people going for is to complete a poster presentation or develop a full research publication. So at this point, I want to invite a couple of our research fellows that are going to share just a few words about their experience in the research fellowship so that you can see what they went through and how this process developed for them. So Sonalika, if you could please make sure that they are ready to share their slides. And as we're waiting for them to share this, I want to ask and address some of the questions that are in the chat. Okay, so let's go through these questions, and Sonalika, please let me know when the next research fellow is ready. Okay, so is it possible to go for 3 or 6 months? Yes, we will address the questions of registration just a little bit. What is the pricing? We will go through the pricing again in a little bit. We also have some, so there's not really a price to the research project itself. There's a whole program that you can register for, and that gives you access to the mentorship, gives you the access to the experts that will provide you with feedback on your project, and also gives you access to the training and the big data analysis tools. Okay, so just so that everybody can see how to actually register, I'm going to just share a link here real quick. So if you go to the top right here and you click on research, okay, right here, this is where you complete the registration. Okay, so if you're interested in the fellowship program, this is where you go. This is where you complete the registration. If you have questions about pricing, this is the pricing. You can switch between 3 and 6 months, and then you'll be able to choose which option is best for you. If you're interested in a scholarship and you come from a low-income country, or you have a special need, there's also a scholarship process that we have that you can use to apply for a scholarship. We only will give a number of scholarships, so the earlier you apply, the better.